Hello friends, so let us start with our next topic, that is secondary active transport. In the previous topic we have discussed about primary active transport, which uses ATP as a source of energy to transport the substance from lower concentration to higher concentration. In this session we will be learning about secondary active transport, which again of course transport the substance from lower to higher concentration, but it does not use ATP as a source of energy. Remember, this is the very important consideration for secondary active transport. That is, number one, substances are transported from lower concentration to higher concentration, but the second point is no ATP is used. No ATP is used, but still energy is used. Energy is used in other form. Say for example a potential energy. Now how do we manage to have a transport of a substance from lower concentration to higher concentration, that is against the concentration gradient, without using ATP? The answer lies in the functioning of primary active transport. Let us go with an example first. See, imagine there is a small mountain, or there is a small cliff, or you can say a mountain peak, and a man or a person uses all his energy and carry a big stone on his head all the way climbing up to the mountain, and keeps the stone on the peak of this mountain, on this peak. Now by doing so, this person has used all his ATP. All ATP is used and the substance is transported from lower concentration to higher concentration using all the energy. Now because we have gathered the substance at higher concentration using ATP, this stone at the peak also has developed a potential energy. Suppose if this person has to keep another stone from lower concentration to higher concentration, but now this person doesn't want to use ATP, but still he wants to transport this red stone from lower concentration to higher concentration, you can say from this base to the peak or to the apex. So one simple mechanism would be tying a rope to this red stone, tying the rope from the black stone to the red stone and pushing the red stone downwards. In doing so, the red stone would drop down, but it will pull all the way the red stone on the top. So the red stone will be pulled on the top in exchange of the purple stone or black stone going downwards. In this way, the red stone has moved from lower concentration to higher concentration by using the energy of the previous transportation, previous energy, that is black stone, and this black stone was put at higher concentration or at higher level because of ATP. So in primary active transport we are using ATP to transport substance from lower concentration to higher concentration, okay, but in secondary active transport we are not using ATP to transport the substance from lower concentration to higher concentration. In fact, we are using the energy of the primary active transport, which has moved from higher to lower, but in doing so it has transported our stone from lower to higher. In the similar manner, secondary active transport uses the potential energy which is collected as a result of primary active transport. Let us go with one very excellent example here. See, the example starts with one of the transporter. In secondary transport we can use the words like exchanger, counter transport, co-transport, symport or antiports, which we have written in the previous session. So let us take an example of an exchanger. An exchanger which exchanges sodium and calcium ions. called as NCE, sodium calcium exchanger, or NCX, that is sodium calcium exchanger. So this NCX, sodium calcium exchanger, it is present on a cell membrane. We have a NCX presenting on a cell membrane, and this exchanger, it transports sodium. Remember, this exchanger, it transports sodium from outside to inside in exchange of calcium from inside to outside. Now you will be wondering which is the transportation, lower to higher in this case. In this case, the calcium is transported from lower concentration to higher concentration. Calcium is transported from lower concentration to higher concentration. But this calcium is not moving all alone. The movement of calcium from lower to higher is actually achieved by the sodium movement, which the sodium is moving from higher concentration to lower concentration. So using sodium's help, that is sodium is moving from higher to lower, which is natural movement, we are transporting calcium from lower to higher concentration. So in secondary to transport, we are not using ATP, still the substances are transported from lower concentration to higher concentration. But instead of ATP, we have used the energy of movement of sodium ions. Now how this energy of sodium ion movement is achieved? This sodium movement or energy of sodium movement is achieved because of primary active transport. See here, what happens? In primary active transport, we have discussed an example of Na plus K plus pump. Now what does the Na plus K plus pump does? The sodium potassium pump, it transports 3 sodium out of the cell. It transports 3 sodium out of the cell using ATP as a source of energy. And the same pump transports 2 potassium from outside to inside using ATP as a source of energy. Now what we have done here is that, as one of the rule, sodium is always more outside the cell. Sodium is always in higher concentration outside the cell. So let us write that sodium we have always in higher concentration outside the cell. But what we have done here is that even though sodium is at higher concentration, the pump is pushing more sodium out of the cell, increasing further concentration of sodium in the ECF. So sodium is already more outside and we are pushing more sodium out. So in this manner, the sodium has gained a very strong force to move inside the cell. See, there is already a crowd gathering at one place and we are pushing more crowd in the same direction. So when that crowd which is already gathered and pushing more people in that area will generate a force in that crowd, that crowd will definitely try to pull back in from the place where it has been pushed out. Similarly, sodium is already more outside and we have pushed more sodium out. So the sodium outside the cell has gained so much force that it tries to come forcefully inside the cell. And this force is further increased because sodium concentration inside the cell is decreased. So a very high gradient is created because sodium is more outside and it is pushed outside, and the interior, the ICF sodium, is reduced. So this force of sodium to move inside the cell is utilized by the secondary active transport. The secondary active transport uses the sodium energy, or you can say sodium linking. It links the transporter to a sodium movement and transport another substance from lower concentration to higher concentration. So sodium always moves from higher to lower. In secondary active transport, sodium always moves from higher to lower, and using this energy of sodium ion movement, we transport another substance from lower concentration to higher. I'll repeat once again. In secondary active transport, the sodium always moves from higher concentration to lower concentration, and using this energy of sodium from higher to lower, we transport another substance from lower concentration to higher concentration. If sorry, if this sodium movement is coupled to a substance which is moving in opposite direction of sodium, like in this case sodium is moving in this direction and calcium is moving in opposite direction, such type of movement is called as exchanger or co-transport. Such type of movement is called as an exchanger. Remember, an exchanger or counter transport. An exchanger or counter transport. Exchanger or counter transport, where one substance moves in and the substance moves out, opposite direction. Both the substances are moving in opposite direction. So exchanger or counter transport. Whereas we can have another example where sodium and the other substance, both of them are moving in same direction, but sodium is moving from higher to lower, but the other substance is moving from lower to higher. The other example is SGLT, that is sodium glucose linked transportation or sodium glucose co-transporters. SGLT is one of the excellent example, and it is always one of the favorite topic for all the students and teachers to appear during exam. So SGLT, what is SGLT here? SGLT stands for sodium glucose co-transporter, or some books mentioned as sodium glucose transporter, and some mention as sodium glucose linked transporter or sodium linked glucose transporter, whatever you say it. But the one thing is, the glucose transportation is linked to the movement of sodium ions. Now, where does this SGLT is present? Primarily, we find two types of SGLT, that is SGLT1 and SGLT2. SGLT1 is very commonly found in intestinal epithelial cells or intestine. In the intestine, the SGLT1 helps in the transportation or absorption of glucose and galactose. SGLT1 helps in the absorption of glucose and galactose, whereas SGLT2 is found in nephrons, PCT of the nephrons, renal tubular cells. So proximal convoluted tubule or tubular cells of PCT, we find the SGLT2. So it's very easy to remember. We eat food at the first hand and then we go for urination. So SGLT1 is in the intestine where it is going for absorption, and SGLT2 is in the kidney where it occurs for reabsorption. In both the cases, glucose movement occurs from lower concentration to higher concentration. Always remember. And because both of them, sodium and glucose, are moving in same direction, hence it is called as co-transporter or symporter. It is called as co-transporter or symporter. Let us try to understand how does it work. I'll take an example of the renal tubular cell or cells of proximal tubules. So say this is a proximal tubular cells or renal tubular cells of PCT. We have a nucleus here. On one side of this renal tubular cell is an apical membrane which is doing the function of filtrate movement. So filtrate moves in this lumen. So this becomes the lumen. This becomes the lumen of the PCT, where we have the movement of filtrate and the tubular cells of PCT. On the other side of the tubular cells, we have blood vessels. So we have tubular cells and we have blood vessels. So it is one of the very interesting mechanism now. When the filtrate is filtered from the glomerulus in the kidneys, in the nephron, the glucose and sodium and many salts, they are getting filtered. This filtrate, which is moving from the lumen, this filtrate contains many important and useful things and also many waste products. all the useful products need to be reabsorbed, because which product does not have to be reabsorbed. So here we have, suppose, sodium and glucose. We have sodium and glucose which we need to reabsorb, because sodium and glucose both are required. So all the substances whichever we are reabsorbing, they go from lumen to cell and cell to blood. Lumen to cell and cell to blood. So going back to blood, it goes back to the body. So in this way the tubular cells, they reabsorb the substances from lumen to cell, that is from filtrate. It takes the useful substances and these substances are given to the blood. So glucose is moving from lumen to cell and cell to blood. Sodium also moves from lumen to cell and cell to blood. How does this happens? On the apical membrane, that is this side, we have SGLT. We have SGLT presenting on the apical membrane side, SGLT2, which transport the sodium and glucose from lumen to cell. As I told you, in SGLT, both sodium and glucose, they move in same direction. Hence they are called as symporter or co-transporter. They are moving in same direction. Sodium and glucose both are moving in same direction. Sodium and glucose both are moving in same direction, that is from the filtrate and lumen end to the cellular end, in same direction. So from the apical side we have SGLT2, and on the basal lateral area where the cell is connected to the blood vessel, we have GLUT2. GLUT2. We have discussed GLUT2 in our previous session of facilitated diffusion. So GLUT2 transport glucose from higher concentration to lower. The GLUT2 is transporting glucose from higher concentration to lower concentration, whereas these SGLT transports sodium. I repeat, the SGLT transports sodium from higher to lower, but glucose is transported from lower to higher. Glucose is transported from lower to higher using the movement of sodium ion, which is transported from higher to lower. So using sodium's energy, we are transporting glucose, and they are moving in one direction, hence co-transporter. So sodium is transported, and along with the sodium, glucose is also transported. So using sodium's help, glucose is transported from lower concentration to higher concentration and in the cell, and from the cell it is transported to the blood from higher concentration to lower. In this way, the distribution of glucose in the tubular cell, always remember, it is as lumen to cell low to high and cell to blood high to low. So lower to concentration to higher concentration and higher concentration to lower concentration, lumen to cell and cell to blood. I repeat, glucose gradient is lower concentration to higher concentration from lumen to cell, and from cell to blood it is from higher concentration to lower concentration. That's why from lumen to cell we are using SGLT, and from cell to blood we are using GLUT2. So this is an active transport and this is a passive transport. This is the very excellent example where two transporters are working and they are trying to achieve the reabsorption process of glucose. The question arises here, how do we achieve this sodium movement? Again, because it's a secondary active transport, a secondary transport never works without a primary. So this sodium movement from higher to lower is only possible because on the basolateral side we have sodium potassium pump. We have sodium potassium pump. Sodium potassium pump transports sodium from inside the cell to outside, causing a very low sodium gradient inside the cell. I repeat, sodium potassium pump transports sodium from inside to outside the cell, causing a very low gradient of sodium ions inside the cell, and this low sodium gradient inside the cell causes the sodium to move from lumen to cell, and this movement of sodium is also been you can say combined to the glucose transportation. So the secondary active transport always needs primary active transport for its functioning. It is not always that the sodium is the only one which is used for the exchanging purpose. The other molecules or ions are also being used. For example, we have chlorine and bicarbonate exchangers, which are called as chloride bicarbonate exchangers or anion exchangers, because both of them are negatively charged ions, so it's an anion exchanger. So we have in certain cells, like we have in RBCs, we have chloride bicarbonate exchanger, which exchanges chlorine against bicarbonate. Here the chlorine is moving from higher to lower, but bicarbonate is moving from lower to higher concentration. So in this way, remember, in secondary transport, one substance moves from higher to lower, and using the energy of that substance, the other substance moves from lower concentration to higher. Okay, so this is the first very important thing we are supposed to remember in secondary transport. This is all what we have learned about the passive and active transportation. In the next session, we'll be learning about osmosis and body fluid compartments. Till then, thank you. See you. Bye-bye.